Hello everyone, and welcome back to our biochemistry series. Today we are diving into part 2 of our exploration into gastric secretions. I am Dr. Muzammil Liaquat Ali, Assistant Professor of Biochemistry. In our previous session, we covered the basics of HCl production. Today we focus on the push and pull. What triggers these secretions and what shuts them down. By the end of this session, you will be able to confidently enlist the various stimulants and depressants of gastric secretions. Most importantly, we will describe the clinical significance and physiological effects these substances have on the body. For those following along with their textbooks, today's lecture aligns with Lippincott's Biochemistry, Chatterjea, and Mushtaq's textbook. I have also integrated key points from standard lecture notes to give you comprehensive overview. So let's start with the stimulants of the HCl production or secretion. The very first one is parasympathetic control. First, we look at the vagus nerve. Vagal stimulation is a major player. It releases acetylcholine, triggers gastrin release, and simultaneously decreases somatostatin, an inhibitor. This creating a perfect environment for secretion. Next one is the histamine, or the cholinergic influence. Because of this, cholinergic drugs like pilocarpine act as powerful stimulant for our gastric glands. Next is histamine. Arguably the most powerful stimulant for parietal cells. When histamine is active, the gastric juice produced is exceptionally rich in HCl. Next is gastrin, released from G cells. Works in two ways. It acts directly on the parietal cells receptors and indirectly by stimulating histamine release from the enterochromaffin-like cells. Next is the calcium levels. Interestingly, hypercalcemic states like those seen in hyperparathyroidism can also lead to increased HCl secretions. Let's discuss additional stimulants like lifestyle factors. Small doses of caffeine and nicotine are known to kickstart the gastric glands. Dietary proteins is the next. High protein meals increases gastrin liberation, which makes sense as the stomach prepares to digest them. Next is the alkali paradox, or alkalis. Sodium bicarbonate, NaHCO3, is in small doses actually increases secretion because the rise in pH in the pyloric antrum stimulates more gastrin release. Next stimulant is the medication and the stress. Drugs like NSAIDs increase acidity by inhibiting prostaglandins, which normally decreases acidity. Furthermore, chronic psychological factors like anger and anxiety are clinically linked to higher HCl levels. Now, let's discuss the depressants of the HCl secretion. The off switches. The very first one is the fats, right? Ingesting fats triggers the release of enterogastron from the small intestine, which inhibits gastrin and slows down stomach motility. Next is the acids, right? When the environment becomes too acidic, the body naturally inhibits further gastrin release as a feedback mechanism. Next is the pharmacological blockers. We can also use drugs to intervene. Parasympathetic drugs like atropine and H2 blockers, such as cimetidine and ranitidine, are highly effective at decreasing HCl levels. Let's discuss the advanced depressants. Proton pump inhibitors is the next, or the PPIs. For more direct action, PPIs like Omeprazole target the hydrogen potassium ATPase pump directly, as we discussed in our lecture series 1, right? Next is the secretin. Finally, the hormone secretin plays a role in decreasing overall secretions of the gastric environment. Thank you for joining today's session. If I missed anything from your lecture style, just let me know what I got wrong and what else I need to include in your lecture series, right? Thank you very much once again.